Good evening. In our previous class, we stopped our discussion with the T cell development. We completed the process of T cell effector responses, wherein we looked at so for the T helper cells, the effector response is ideally release of certain cytokines, which contains information regarding which cell needs to come up, say, from the innate immune system, or if there is a killing function that has to take over, or if there is some phagocytic function that has to be enhanced for clearance of pathogen, whatever is the information that the immune system wants to convey to the body, it's conveyed in the form of certain cytokines released from the T helper cells. We also looked at the different subtypes of cytokines, and we saw how cytokines are exactly, we can see that cytokines are what is mediating the communication between the cells of the innate as well as the adaptive immune system. On the other hand, we have the TC cells or the CD8 positive cells, which mediate the killing function of the pathogens, and that was being brought about by two pathways, either the fast pathway where there is binding of fast and fast ligands. This initiates the this initiates the synthesis of apoptosis related genes, and eventually the cell is triggered for death. The other pathway possibly is the perforin or the granizyme pathway, wherein there was certain sort of adhesion, a temporary adhesion was being formed between the target cells and the CTLs. At the site of contact, there was release of certain cytotoxic components, and how exactly they are mediating the killing function. They are leading to formation of some pore-like structures on this membrane of the cell, target cell, and eventually through it we have passage of certain other molecules, the granizymes, which trigger the cells for apoptosis. So that is how this is the response of the humoral immune system and of the cell-mediated immune system. And prior to this, while we were discussing our B cells, we saw that the humoral response is primarily mediated by the antibody molecules, right? Okay, so today's class we are going to actually bring about compilation of the information that we have been learning about in as two separate entities, right? So we discussed that uh, there's two bifurcations of the immune system: the innate immune system, the adaptive immune system. For the innate immune system, that have we had certain barriers, some physiological, some physical, some cellular barriers. Those things were compromised by the innate immune system. And in the adaptive immune system, we further divided it into two components, right? The cellular branch and the humoral branch. So in today's class, we will see that, and we also discussed in some of the classes that it's not that these two immune systems are going to function separately. They need to communicate among themselves to bring about effective clearance of the pathogens. Now, in today's class, we will see that we do have killing mechanisms which are also performed by some cells which belong to the innate system. And we will also see that there are certain, like the antibody molecules that could bring about some sort of clearance of pathogens by different mechanisms. We will also discuss those mechanisms, that how production of antibody is okay, but how exactly that antibody is mediating its function, that is what we are going to discuss today. And we will also see that there are certain the antibodies alone are not sufficient to bring about the lysis, say, of the pathogen, but they do require some complementing proteins named as complement proteins to bring about its effector response. Okay, so to begin with, since we have discussed the killing mechanisms in previous class, we will we will see that there's another cell in the body which also has the killing which can also perform the killing of target cells. So that cell is the natural killer cells. Now, if you remember from the discussion where we discussed about the cells and how the different lineages from where they come from, so there was one hematopoietic stem cells. They first divided into two different types of cells: the myeloid progenitor cells and the lymphoid progenitor cells. Essentially, all your lymphocytes come from the lymphoid progenitor cells. But along with the B and T cells that are being formed from the lymphoid progenitor, from the same precursor cell from where we have the T cell forming, there is another cell that was formed, which is the natural killer cell. These cells, like the name suggests, they must be killing off the target cells. And it was also very experimentally, it was noted that the killing mechanism is somewhat very similar to the killing mechanism of the T cells. So there are some similar features of natural killer cells with the T cells, and there are some features which are distinct between the two classes. So if you look here, so the first line of defense against a virus infected cell is actually the natural killer cells. So we say that we saw that the intracellular pathogens are primarily taken care of by the T cells because the humoral component can only take care of the extracellular pathogens. But T cell response, it's a part of the adaptive immune system. And adaptive immune system, if you recall, has a lag period. So to avoid what prevents the pathogen from causing any much harm to the body during this lag phase is the first line of defense, which is the natural killer cells. And again, first line of defense is it refers to the innate immune system. So natural killer cells are a part of the innate immune system. So the natural killer cells, they form the first line of defense and control the viral replication during the lag phase of the immune system, of the adaptive immune response. Now, they share some features with T cells, such as the progenitor is common, but as I said, that there are certain things which are also distinct. They do not develop in the thymus. They continue to develop in the bone marrow, and from the bone marrow, they get into the circulation, wherein they can be activated by certain cytokines, and in turn, they can also produce some cytokines, primarily the interferon gamma. Interferons, again, in cytokines part, we saw that they are primarily responsible for the viral defenses. So, interferon gamma is produced, which also imparts a state of virus resistance in the nearby cells. And this interferon gamma also signals for the Th1 differentiation. How does it do it? It says that interferon gamma says to the macrophages that they need to produce IL-12, and this IL-12 gives a positive stimulus for Th1 differentiation. We also talked about it that there are two sets of cells, Th1 and Th2, and both the cytokines produced from either of these cells actually give a positive signal for further differentiation into the same cell type, and at the same time negatively signals the differentiation of the opposite cell subtype, right? And how do they take care of the virus? They bring about killing in the virus, they all by the mechanism of fast mediated pathway. So here again we have fast ligand, which is present on the surface of the natural killer cells, and they kill the target cell that bears this fast receptor on its cell surface. And very similar to the T cell, they have certain granules, but distinctly from the T cells, the granules are constitutively present. So if you remember, the granules were initially not present in the CTLs, and it's only upon um, but the granules in T cells, so they were not constitutively present. It is only after they receive some sort of stimulus from the in form of cytokines from the T helper cells or from the B cells that they have the granule expression coming into the cell, right? But in the natural killer cells, the granules with perforins and granozymes are constitutively present. So very similar to T cell, they can bring about killing by either of these pathways. And again, comparison. So they do not have the CD3, they do not have the TCRs, which are very specific to T cells. They do not have any sort of MHC restriction. They do not need stimulus from MHCs. And there is no memory response. If there is a pathogen encounter, for the second time, the response is very much similar to how the response is if it was a primary encounter. Now, if there is no MHC restriction, there is no CD3, how exactly is the signal being communicated? So it has been found that the NK cells, they can distinguish between the self cells and the altered cell by using two sets of receptors. The first set of receptors are the ones that can give an inhibition signal, and the opposite, the contrary, they have the activation signal delivering receptors. So these are two classes of receptors that are present on the NK cells. Now, in inhibition signal delivering receptors, they can either be some C-type lectin inhibitory receptor, or it could be some killer cell inhibitory receptors. Just some receptors that if these receptors get bound, if they receive a signal, the cell will be told that you are not supposed to kill this particular cell type. So again, so for killing, there has to be an interaction between the two cell types. Now, if there is an NK cell and it is getting bound by a target cell, the target cell can have certain moieties decorated on its surface, which can bind and give a signal to either the inhibitory set of receptors or the activatory set of receptors. If the binding of the receptor is to the inhibitory set, the NK cell is instructed that you are not supposed to kill this particular cell. But if the activation signal is activated, then the target cell is taken for killing, and you will have the release of perforins and granizymes from the granules between this contact site, and in a very similar mechanism, the killing function will be carried out. Now, which all receptors actually constitute, if there is binding of these receptors, there is going to be an inhibitory signal. It is the C-type lectin inhibitory receptors and the killer cell inhibitory receptor. And if the activation signal, those are getting delivered by C-type lectins, CD2, and the Fc gamma 3 receptor, which is also known as the CD16 molecule, right? So C-type lectins, so lectins are something that binds to the glycoproteins or the carbohydrate moieties on the surface of some pathogens, right? So maybe on the pathogens, or maybe some processing that displays some cells that can express some glycoproteins on its surface if, say, there is an infestation of the pathogen intracellularly. Now, the activation signals are further stimulated by some cytokines again. So if there are these cytokines present here, the receptors which can deliver activation signals is upregulated. So these cytokines essentially tell the NK cell that there might be a requirement of your activation. So you upregulate these receptors so that if a target cell that can be bound by these receptors is present, the chances of NK cell encounter to this target cell is enhanced, right? Now, how do they identify a cell which is having a problem? So, NK cells are actually capacitated to recognize cells that have aberrant MHC molecules. So, what happens? It's your cancer cell or the virus infected cell. The MHC1 expression is downregulated in your tumor cells, as well as if there is a intracellular viral infection, right? So binding of this MHC class 1 occurs with the inhibitory set of receptors. So if you have a normal cell, there will be normal display of class 1 MHC molecule. The stimulus is given to the inhibitory cells, inhibitory cell receptors, and there is uh, there is no activation signal. So because of this binding of the negative, this inhibitory signal, there is no killing of the target cell that is getting mediated. However, if there is a virus infected cell or if there is a cancer cell, if you look here, the MHC molecule that was present is no longer there. So the negative stimulus that the NK cell was receiving so far, it is not going to receive it, and only the activation signal is getting it's the only signal that the NK cell is receiving, and that's how the killing function of this particular cell is taken forward. So another type of cell that we discussed is the NK cell, which essentially is a cell of the innate immune system, but can also bring about killing function. So there is a arm of innate immunity that can mediate killing. We already discussed the arm of adaptive immunity that can mediate the killing of target cell. Now there is also a arm of humoral immunity, the humoral component of the adaptive immune system, that can also mediate the killing function in a particular way. So this mechanism involves the antibody molecule, and that is why the phenomena is known as antibody-dependent cell-mediated cytotoxicity. Antibody-dependent And because there is ultimately, so antibody molecule in itself cannot get inside a cell or kill a particular cell. Antibody molecule is there to specifically mark a cell type. So antibodies are very specific and they are going to recognize the antigens present on the pathogens. Now, if there is a requirement of killing of this particular cell, there is going to this binding of antibody to the pathogen is going to signal certain other cells of the innate immune system to come and bind to the antibody molecule, and eventually the clearance or the cytotoxic function will be mediated by those cells that come and bind to the antibody molecule. So if you imagine this, there's a pathogen, we have the antibody molecule binding to it. The binding of the antibody on the pathogen molecule is highly specific. Now there is another cell here which has some sort of receptors for the constant region of the antibody. So this cell with receptors for the constant regions of a particular antibody, say IgM subtype of antibody, it comes and binds here. And see, this is a phagocytic cell. So this is a phagocytic cell. Binding of it to the antibody molecule is significantly going to enhance the phagocytic capacity of the particular cell. And this cell is then going to engulf this pathogen mediated by so the recognition part of the cell to the pathogen was mediated by antibody. So in one way, the innate immune system is somewhat specifically recognizing a pathogen. However, the specificity component is being achieved by participation of an antibody molecule, right? Okay, so the antibody mediated, mediated by cells that have receptors for the Fc region of antibody. Which all cells can actually have Fc receptors? It could be the NK cells that we just discussed, the eosinophils that come from the myeloid progenitor of cells, the macrophages, mast cells, or the monocytes. So these are all cells from the myeloid progenitors, right? So if look at this diagram, so it discusses all the cell types that actually have Fc receptors for your constant region of antibody. So initially we have a target cell which is displaying some sort of antigen, say, and which class of MHC displays okay. So no class of MHC. MHC is not involved for antibody recognition. The target cell has some pathogen on its surface, and this has some antigen on its surface, and this antigen is now first getting recognized by the antibody molecule in a specific manner. So all these antigens are now, if you look here, are being bound by antibody molecule. Now the Fc region of the antibody molecules, based on which type of antibody is bound here, there can be different cells that can come and bind this Fc region. So let's start with macrophage. If there is binding of macrophage to the Fc region of the antibody molecule via the Fc receptor present on the surface of this macrophage, the phagocytic activity of this cell is enhanced multiple times, and eventually it is going to engulf the target cell and bring about clearance of the target cell. If the binding is by the NK cells, the Fc receptor of the NK cell binds the Fc region of the antibody. At the site of contact, the NK cell releases certain cytotoxic molecules such as the lytic enzymes, perforins, TNS, and the granizymes. And this is going to be released at this site of contact. And eventually, we will have pore formations that is going to kill the cells. Similarly, for the neutrophils, the binding of neutrophils to the Fc region of the antibody releases a lot of lytic enzymes. So there's going to be enzymatic death of the target cell. And if you look at eosinophils, eosinophils have a lot of lytic enzymes and porphyrin molecules. And if there is this binding, then the lytic enzymes and porphyrin molecules are going to kill the target cells. So here, if you see that by themselves, the eosinophil molecule could not recognize the target cell or bring about some sort of killing. There is a dependence on the antibody molecule, and that's why this entire process of cell killing, this phenomena is known as antibody dependent cell mediated cytotoxicity. Okay. So this makes it very clear to us that the innate immune system has certain killer cells. We have the humoral branch, which can also mediate this killing by becoming a via media in the process of it, and we also have the cells of the cell mediated immune response of the adaptive immune system, which can definitely bring about the killing function. So with this clear, it is nice to have a look at the overview of where we have actually started with the entire system, what all we have already discussed, and what remains to be discussed further. So, quite a few things we have already discussed for our immunology concept, right? So, immune system. So, immunology is the study of the immune system. This immune system, we divided it into two arms: the innate immune system and the adaptive immune system. Now, for innate immune system, our discussion was somewhat straightforward. We discussed that there is some barriers, physiological, so wherein we discussed about the temperature or the pH, the chemicals that are present in your body. All of those are called are incorporated under the heading of physiological barriers. We then have the physical barriers. So these were your skin or the gut lining or the respiratory tract lining. So any sort of lining that constitutes a physical barrier. We have the cellular barrier where the phagocytes, the eosinophils, the neutrophils, basophils, all sort of cells of the innate immune system that we discussed. So which all cells? It could be macrophages, monocytes, eosinophils, basophils, and neutrophils. And of course, we just discussed the NK cells. So these are all players of the innate immune system. Now, there were also some sort of soluble components that we said that can also form a defense. And in the soluble components, we have not discussed this, but in today's class, we will look at it. So these were some chemokines, the cytokines that we are talking about. So those are soluble components from the adaptive immune system. But very similarly, the soluble components of the innate immune system is the complement proteins. Okay. In the adaptive immune system, there is something called as the cellular immune cell me or cellular or say cell mediated response and the humoral response. So humoral response, this involved the B cell produced antibodies And what all functions the antibodies can mediate? Okay, let's just You've come quite far. So we begin our discussion, and I'm just trying to keep the same thing here so that we know what all we have discussed so far. So they could either neutralize the antigen. We will have a look in today's class how that what neutralization of antigen means. They could cross-link several antigens. So maybe one pathogen molecule can display different types of antigens, and we have different antibody that can bind to the different types of antigen. But cross-linking can be brought about when, say, in this sort of way, two antibodies molecule, if they are coming together because of one common antigen, this cross-linking mediates a lot of important biological functions. Alternatively, they can activate the complement system. So again over here, so what is important for us to understand is the antibody molecule in itself only has the potential of binding something specifically. But the response that is mediated is because of the FC region of the antibody that gets bound by certain other cells. So these cells, or maybe some other proteins that bind to this FC portion of antibody. So either some cells or some complement proteins have to bind to the FC region of antibodies in order for any sort of effective response to come up from the antibody molecule, right? At the same time, in cell-mediated immune response, we have two sorts of what T cells, which can be divided into two subtypes, the T helper subtype and the cytotoxic T subtype. So T helper subtypes, we also divided this into multiple types. So Th1, Th2, T follicular, T rex, right? Even if I'm missing on something, and then we saw that this TC in eventual set of differentiation, it becomes the CTL. This bring about killing function, whereas the function of this is to produce different cytokines. Okay. So we have discussed, so if one thing that is actually, then that means that, and which all region can the cytokines communicate to? The cytokines can instruct the cells of the cytokines can instruct the cells of the innate immune system. The cytokines can instruct the T cells. The cytokines can also instruct the B cells, right? So one instructing molecule which is common to to both the adaptive and the innate immune response is the cytokine. So cytokines is a common instructive molecule, and a common effector molecule is the complement system, because complement system in itself is more of a part of the innate immune system, but then there is one pathway of complement activation which can be dependent on the antibody complex, antigen antibody complexes. So effectively they are mediating an effective response of the adaptive immune system. And it is also, so one thing that, so we've been briefly talking about it, is that, so the role of macrophages is to phagocytose, of monocytes, basophils, and neutrophils is to produce lytic substances. Mm, okay. They can produce lytic substances, or say inflammatory substances. So inflammation, that in a way talks to the, it contributes towards the physiological barrier, so the temperature part, or the the release of certain other, so inflammate in the process of inflammation, if you remember, we had certain sort of chemokines, which again is a type of cytokines. So this is a complex network of multiple effector responses that is being, so if you look here, if you can bifurcated. So there are different arms of it. But if you keep drawing these lines at what is happening by one system, you will see that the effector response is also triggering some effector response of the another arm of the other bifurcation that you have just drawn. So the idea is to appreciate the overall interconnectedness of the systems, right? Okay, so I'm sure I must be missing something in here, because the more you look at it, the more you will be able to fill it. So once you all have the notes, I would appreciate if either of you could fill in more details onto this diagram, and maybe share it with others, or just keep it for your reference, so that in one go you would be able to see every topic that we have discussed in the immune system. Okay, so we have touched upon almost every part of it. The only thing that we have not discussed in much of a detail is the complement system. That is the main topic of discussion for today's class. So in complement system, how was the let's just discuss the discovery part of it. The complement system was discovered when Jules Bordet performed an experiment wherein cheap antiserum, so antiserum means the serum that has some sort of antibodies against the bacteria Vibrio cholerae. So he isolated the antiserum from the sheep which had bacteria Vibrio cholerae infections. So there must be some sort of antibodies in that serum. So when he took that serum out and he incubated it in vitro condition with the bacteria, he noticed that there was bacterial lysis. So it was easier to expect that there must be the antibody molecules there, and they are bringing about the lysis of the bacteria. And if we heat up that serum, the bacterial lysis was not happening. So people thought that, okay, that's okay, might be the antibody is getting denatured, because antibodies are glycoproteins, right? Immunoglobulins, so not glycoproteins, immunoglobulins. So the bacteria it might be the immunoglobulin structure, the folds and all might be getting compromised, and there is no bacterial lysis that is happening. But a fresh observation came up that after heating, if he could supply some fresh serum which did not have antibody, and alone it could not bring about the bacterial lysis. So if you just take this fresh serum from, say, a sheep which has never got infection of Vibrio cholerae, there will not be any antibody in its serum. And how to verify it? That if you just incubate it with the bacteria in vitro, there is no bacterial killing. But if this antiserum was subjected to heat, and there was no bacterial lysis. But if there is a supplementation of fresh serum with this heated antiserum, the bacterial lysis was restored. So now the confusion came up that what is there? So this means that there must be two components. One that is heat labile or sensitive to heat, which is getting denatured in because of heating. And there must be another component which is not as sensitive to heating but requires this heat labile component, because if there is heat, the heat labile component is not present, and this alone could not bring about bacterial lysis. But this other component was not heat labile. So now if we take this heated O and we have a fresh serum which did not have antibody, so one component had to be antibody as per the guess, but then what is this other component? So there he isolated whatever was there in this fresh serum, and maybe some sort of fractionization studies came up. Because of that, it was identified that the antibody could survive the heat. It is the complementary molecules. The name complement was actually termed by Paul Ehrlich. So independently, he was also conducting some sort of similar experiments and approached a similar result. Now, in those results, because he would say that there is a complementing factor to the antibody that brings about the bacterial lysis, he named them the complement proteins. And so Jules said that they are responsible, the complement protein is responsible for the lytic activity, because if it is absent, if it gets denatured, there is no bacterial lysis, and antibody alone cannot help the process. Now, what is complement protein? So, a complement proteins was then identified as an important effector arm of both innate and adaptive immune system. So, not just the adaptive immune system, we will just have a look at it, that the innate immune system, a lot of innate immune response is also mediated because of the complement proteins that are present. Okay. So what all effector functions can be brought about by complement proteins? It could be cell lysis, it could be clearance of immune complements or complexes, opsonization, viral neutralization, inflammatory response, and solubilization. So opsonization, viral neutralization, inflammatory response, all of this, if you see, so these are also broadly all the effect that the antibody molecule can also bring up. So this therefore is important for us to appreciate that antibody molecule, most of the function that is coming up because of the participation of antibody molecule is mediated by the complement activation. So if apart from the ADCC function that we just saw, most of the effector responses coming up from the antibody presence of antibody molecule is mediated by complement proteins that help the effector response of antibody molecule. Oh, so just a schematic representation here. So the lysis of it. So this is a target cell, and binding of the complement protein is going to cause a lot of cell bursting, and the components of the cell is coming out. So this brings about cell lysis, opsonization. So what this process of opsonization, the word might also be new to you all. But what does it mean? That it means that there is some sort of coating that is happening on the pathogen molecule. So if you look here, the bacteria is coated by the complement molecules. Because of this complement coating on the bacterial molecules, it is easier for the phagocytes to now engulf the bacteria, because the phagocytes have some receptors for the complement protein. So we just saw in ADCC that there are some receptors on the phagocytes for your FC region of antibody. Similarly, there are receptors on the phagocytes for the complement protein. So these are complement receptors. Now, complement proteins also mediate the inflammatory response. So, what happens in inflammatory response? We talked about the fact that there is the phenomena of diapedesis happening. So, diapedesis or extravasation, wherein the lymphocyte first gets marginalized with the endothelial lining and eventually squeezes out through the endothelial lining. The process is called as extravasation. Now, how does this leukocyte know that I have to come out from this portion, or this is the site of injury? we have certain complement proteins which are mediating this extravasation process. So we will look at it. So there is something called as anaphylatoxins. So some of the complement protein or the some of the fragmentation products of the complement proteins are anaphylatoxins, and they instruct the lymphocytes to get marginalized and extravasate at the site of tissue injury. And they can also bring about, if there is binding of some complement proteins to the receptors present on the eosinophils or the basophils, the granules present in these granulocytes can also get degranulated, and a lot of lytic substances or the enzymes can be released at the site of injury. So, by the binding of complement proteins to receptors present on specific cell types, different inflammatory responses can be activated. Apart from that, we have so binding this, so there might be some soluble antigens that come up. The soluble antigens are bound by the antibody molecule. That much was the job of antibody molecule. Now just binding is not going to clear the antigen antibody complexes, right? So we again have some sort of complement proteins binding to these antigen antibody complexes. This, in a way, pulls the entire complex towards the phagocytes. How? That these complement receptors bind to the complement protein, and say like a magnet, it's going to attach. So this complement instructs the movement of these immune complexes towards this phagocytic groove, and eventually the phagocyte is going to take it up. Or the other possible way is these complement proteins, they can also bind to the RBC. So we will also have a look at it. The RBCs also have the complement receptors. So the anti-immune complexes via the complement proteins get bound to the RBC molecule. So all the antibody complexes also get bound. And where do these, so RBCs only have a life cycle of 120 days, and eventually they are pulled to the spleen. So in over there, the RBC is when the RBC is taken into the spleen, along with it, all the immune complexes also reach the spleen. There, the binding of the FC receptor, the complement receptor, and the complement protein is stripped off, and eventually the phagocytes, like very similarly as here, they do phagocytic clearance of these immune complexes. Okay, so that's just an overview of all the pathways that are mediated by the complement proteins, and let's just discuss that how exactly, what is the process of activation of these proteins, and what are the different pathways and the players that are involved in it, right? Okay, so first the components of complement system. So they are constituted by a group of proteins or glycoproteins that can be designated as C1 to C9 in the order of its discovery, right? So it got nothing to do with maybe the position of its participation in the overall pathway. So if one protein was designated, it was discovered first, it was named C1, and the next in line was known C2, and so on. So we have some proteins and glycoproteins designated as C1 to C9 in order of their discovery. So those are not all. So we mentioned that there might also be some that are known by their common name, because it might give you a sense that there must be just 9 proteins, but there are around uh 30 complement proteins that have been identified so far. So C1 to C9 is a number, the order of its discovery, and there are other participants also which are named by the common name, maybe some scientists that who identified, given it some name, and that's how, right? So in a total of around 30 complement proteins have been identified so far. Who synthesizes the complement protein? Mainly they come from the liver hepatocytes. Some other sources of it could be the monocytes, tissue macrophages, or the epithelial cells in your gut, right? The GI tract, gastrointestinal tract. Now, activation pathway. So if you look here, so proteins that are just present in your circulation in themselves, they are not activated. They are in their inactive form, and there has to be activation of one molecule which then activates the next in line. This next molecule then will activate the next protein in line, and that's how a cascade induction happens, right? So just to so what does this cascade means? It means X activates Y, the Y then activates Z, Z can activate the other in line, right? So one after one, there is going to be cascade induction. Now, how is this X activated? Is the first initiator, activation of the first initiator, that could be different for either of the pathways that we just discussed, right? So activation, but let's just for now just say that there is an activation that has happened, and upon activation, what happens is that these components that you look here, they will split into. One is going to activate the next in line, and the other is just going to mediate some other function, right? And as a part of convention, the the fragments are designated with lowercase number. So how are the complement proteins designated? If you look here, they are named as C1, C2, C3, C4, C5, C6, C7, C8, C9. So like that, right? And even if the ones that are known by common names. So let's just say there's a factor called factor B that we will just have a look at. So this there is whole of, say, just let's just take an example, maybe C1 complement protein. Now C1 will cleave into two parts, the C1A and C1B. Now, as a matter of convention, this is how we write, right? By small case letter. Now, this C1B is also whatever is designated by B. So, this cleavage that happens here, it is not a proportionate cleavage. One is always a smaller fragment, and the other one is a larger fragment. The smaller fragment by convention is named as A. The larger fragment by convention is designated with small b, with an exception of C2. So in case of, so I'm not sure how exactly, there always has to be some exception for us to remember, but nonetheless, there is an exception of C2. So C2 is the only complement wherein C2A refers to the larger fragment and C2B refers to a smaller fragment. Just okay. So so the smaller fragment, it mediates, so this smaller fragment is going to diffuse out from the site of its cleavage, and it's going to mediate inflammatory functions. And the larger fragment, it is going to bind to the next target in line, the next protein in line, and in sequence, and eventually bring about cleavage of it. So and activate this other protein. And again, the cleavage, when there happens a cleavage, there might again be two unequal fragments, one being C2A, C2P, maybe just A and B. The A, or so let's not say A always goes, because then that kind creates. So it's always a larger fragment that mediates activation of the next in line, and the smaller fragment mediates inflammatory response. Because there's an exception in C2, the larger fragment is designated with small a, and the smaller is designated with small b. Otherwise, throughout the complement system, the larger fragments are going to be designated with small b. Okay, now these different fragments associate as an enzymatically active component. So what happens is that when one component is cleaved, the active so this larger fragment, it goes and cleaves the next in line. The product of this next cleavage can also the active component. So the larger fragment of the this second protein that got cleaved, that can form a complex with the previously formed larger fragment, and there can be multiple such fragments that can join together, and eventually the joining of some of these fragments together, if they're joined with in the right order and all, they can bring about, and they can themselves become enzymatically active component. And these so enzymatically active means it जैसा enzymes का function होता है वैसा इनका role होगा. और ऐसे components को, ऐसे fragments को, which is being formed by association of multiple such fragments, they are designated with a bar on the letter. We'll just have a look at it, right? Okay, so those are, so this is these are some generalization that you should know, and it can simplify your understanding of the concept that just follows. And to begin with now, how, let's just start with the process of activation pathways. So there are three such pathways. One, the classical pathway, the lectin pathway, and the alternate pathway. So the activation part of these pathways are different. Three such pathways are there, but eventually they terminate together, or they merge together. How? That they all lead to formation of a membrane attack complex. So the formation of membrane attack complex is the terminal consequence of complement activation. And as the name suggests, a membrane attack complex, something goes wrong with the membrane of the target cell, and eventually the cell is triggered for lysis. So this. Okay. So a little complicated to look at, but we'll try to simplify it so that it's easier for you to memorize. Three pathways we discussed. We said that there are three pathways in there: the classical pathway, lectin pathway, and alternate pathway. This is how the activation part of these pathways differ. And this is the common overlap that is there in all the pathways. So let's go one by one. So this is the sequence for your classical pathway. This classical pathway binds antigen-antibody complex. So we say that in a cascade, there has to be one initiation point, and then the next ones are going to automatically follow. So in classical pathway, the initiation point is achieved by C1, one of the complement proteins, binding to the antigen-antibody complex. So since there is an involvement of antigen and antibody, so this pathway, the classical pathway, is an effector response that belongs to the adaptive immune system. And why is it named classical? Because initially this was the first pathway that was identified. So people thought that this is the only function of complement proteins. Later on, the other pathways came up. So since it was the first one, the classic one, this is named as the classical pathway. There might be some antigens which are bound by antibody molecule. Now C1 has a complement protein, which if it binds to the antibody molecule or the antigen antibody complexes, this activates the cascade. So this C1, it gets activated. This is going to cleave the C4 and C2 complement proteins into two fragments, a larger one and a smaller one. So C4 is cleaved into C4b and C4a, and C2 is cleaved into a larger fragment C2a and a smaller fragment C2b. So we'll just come to the further details of it. Let's just have a one go overview first. So it just simplifies your understanding, right? Now these larger fragments, as we were just discussing, that there can be some which come together into enzymatically active component. So C4b and this C2a together, they are designated in this manner, right? C4b, we do not write the C again. We simply write 2a. And if there can be some other molecule which also associates with it, number of it just gets further added to it. We do not write the C again and again. So association of these two fragments as C4b and 2a, this is an enzymatically active component. So therefore, we have this bar. So these are some nomenclatures that are used in the in representation of your complement pathways, right? Now, this C4b and C2a is an enzymatically active component in which enzyme? The C3 convertase. Now, C3 convertase, as the name suggests, it is going to do something. It's going to some sort of, in a way, convert the C3 molecule. The C3 converts in presence of C3 convertase, gets cleaved into one smaller fragment, C3a, and another larger fragment, C3b. Now, because of C3a and C3b cleavage, the C3b molecule that forms, that can again associate with this further and together forms the C4b, 2a, 3b. So this complex that is formed here is again enzymatically active and is known as the C5 convertase. Now, C5 convertase, again, as the name suggests, it's going to cleave the C5 complement protein into C5b, which has to be the larger component, and another complement, the smaller fragment of this C5a. Now, this C5 molecule that forms, it is going to recruit some other complement proteins, C6, C7, C8, and C9 in line. And together, as all of these complement proteins associate one by one in line, it's going to form the membrane attack complex. So we'll just see the visualization of it. So what happened with C5b? C6 joins, C7 joins, C8 joins, and then C9 joins. C9, if it finds an association with all these components, this leads to multiple molecules of C9 joining and becomes a poly C9 molecule. Now, this poly C9 molecule associates on the cell membrane is this manner. So here it forms a pore. So this formation of pore is nothing but this formation is referred to as the membrane attack complex, and this is an electron micrograph that shows the formation of the pores onto the cell membrane. We'll come to the discussion of this in greater detail. For now, let's just follow back to this point wherein we had a C1 molecule. So we have antigen antibody complexes, C1 molecule that binds the complexes. This gets activated and brings about cleavage of C4 and C2. The C3 convertase that forms is constituted by C4b and 2a. This is joined by C3b. So this is a C3 convertase. Leaves C3 into C3a and C3b. The C3b further joins here and forms the C5 convertase. C5 convertase again means cleaving of C5 into C5b and C5a. C5b is joined by C6, C7, C8, C9, and eventually there is an assembly of membrane attack complex. Now, if you look here, now we start adding on to the details. So C1, this is how the C1 molecule looks. The C1 is a complex of three proteins, C1q, C1r, and C1s. C1q in itself is formed by six protein molecules. So these six heads that you see over here. All of these are the C1q molecule. So it's a polymer of six molecules, right? And it is joined by C1r and C1s. So once we have the C1 binding to the antigen antibody complexes, so which part of C1 binds? The C1q. The C1q part binds the Fc region of antibody molecules. Now, different antibodies bind with different, the affinity of binding of C1q to all the different types of, subtypes of, or isotypes of antibodies is not the same. The highest binding or the strongest binding is of C1q with IgM. Now, if we try to bring things together, so this again is a this is again for you to appreciate that how the systems are linked. Which one is the first antibody molecule that is produced in the primary response? The IgM molecule. And IgM molecule is also having a highest binding affinity to C1q molecule. So IgM is also a pentamer, has highest avidity. IgM also has the highest binding affinity to C1q, so it can bring about effective clearance response by involving the complex system. It can bind to multiple antigens and neutralize it. So that's how IgM, that is the first antibody. The body is very smart. It knows that most of the functions, the effective functions that I need to take care of the challenge, can be most effectively mediated by IgM. So let's just produce IgM in the first go. And eventually the switch takes up to the IgG isotypes, right? And of course, the switching of antibody again is mediated by cytokines. But different subtypes, so the idea, let's just come back to complement. So in complement, the most highest affinity binding is of IgM to the C1q component, followed by IgG type G3. So IgG can also be of five different types, 1 to 5. Ig 1 to 1 to 4, 4 ties. IgG3 is of the highest affinity binding in amongst all the IgG types, followed by Ig1 and Ig2. Ig4, IgD, A, and E, they cannot be bound by the C1q molecule. Okay, so once the C1q molecule binds to the Fc region of antibody, the C1R and C1S gets activated. C1R that you see here, so C1R that you see here, it is nothing but a serine protease. So any sort of, so I said complement proteins, so complement system involves complement proteins. And if there is any sort of cleaving happening, it has to be mediated by some sort of proteinases. Any enzyme that cleaves proteins is called proteases. So protease, so C1R is a serine protease. What does serine means over here? Serine means it will cleave at the point where a serine residue is present. C1R is a serine protease, and this is what is actually the activated C1. The C1R cleaves the C4b and C2a. So C4 and C2 that we have been talking about. Okay, so this is what the classical pathway looks like, and just a better visualization and a quick overview of the pathway, okay? So C1q binds the antigen antibody complexes. C1R activates autocatalytically and activates the second C1R, both activating C1s. So just some more details. Just have to remember that C1 is having three proteins together. C1q in itself is a polymer of 6 units. So once the C1q binds the antibody molecules, the antigen-antibody complexes, there is activation of the C1. It is first that the C1R activates by autocatalytically. So it gets activated by self cleavages and then activates the next C1R, so multiple C1Rs, and both then activate the C1S. Uh, Too much of a detail if that looks like, just remember it's a C1q molecule that activates and C1R and S that are the catalytically active ones. Now C1S cleaves the C4 as well as the C2. Cleaving of C4 exposes binding site of C2. So first it is a C4 and then C2 comes in. C4 binds near the surface of C1, C2 binds the C4 forming the C3 convertase. Okay, so this C4b molecule comes here, the C2a molecule comes together in the formation of C3 convertase. Now, C3 convertase hydrolyzes many C3 molecules. There are multiple C3 molecules present here. The C3 convertase has formed. And in its present, multiple C3 molecules get cleaved. So, multiple C3 molecules get cleaved. So this in a way is an amplification step, that we have an enzyme formed here and multiple it acts upon multiple complement proteins. So if there is just one such enzyme, so many C3b molecules can be formed. Now this C3b molecule can mediate some functions on its own, or it can associate back with the C3 convertase. If it associate back, this is known as the formation of C5 convertase. So some combine with C3 convertase to form the C5 convertase. So binding of formation of a C5 convertase allows for C5 to again get cleaved, C5b and C5a. They can have different function on its own. C5b that is formed, so C5b that is formed is joined by C6 and C7. So C5, 6, 7 associates in this manner. This is then joined by C8 molecule. The C8 joining allows for multiple C9 molecule to attach onto the sides of this C5b678. So you have to remember the flow of it, and that only can make it easier for you to understand that what is joined by what. The good part is that the story from here is the same for all the different pathways. Okay. So once we have the assembly of poly C9, this is a pore formation. So this is known as the membrane attack complex. There is transmembrane a channel that allows for free flow of ions and macromolecules. So there can be influx of ions, efflux of ions. There can be leaving of macromolecules. There can be entry of water. And eventually, as a consequence of all of this, the entire osmotic balance of the cell, or the energy or reserves of the cell, all of it is compromised, and that's how the cell is triggered for lysis. Okay, let's now come back here. The other pathway that is here is the alternative pathway. So classical pathway, the alternative pathway. In alternative pathway, here we needed antigen antibody complexes, right? And that leads to formation of C3 convertase, and which is leading to cleavage of C3 molecules. Alternate pathway is triggered by slow but spontaneous conversion of C3. So C3 molecule has a tendency of spontaneously cleaving, but if this cleavage product is joined by a foreign substance, so this C3b, so imagine it like a small number of surveilling army population is constantly forming, and if there is nothing to patrol on, this is going to get cleaved back. But if the army finds an invader, this associates on a foreign substance. This association on foreign substance allows for factor B to join the C3b with the foreign substance. Because of this joining, what forms? So factor B, the factor B also has, if it binds to the so let me just draw it here. So a foreign substance. So what is this foreign substance? It could be some cell, that some bacterial cell, or maybe some altered cell, any sort of foreign substance that the C3b gets a chance to bind to. Now, because of this, the B, so they have actually, I'm not sure, they have kind of missed one step. So this foreign substance is bound by C3b. This is then getting a chance to be bound by factor B, but it's going to cleave it into factor BB, the larger part of it, the larger fragment, and factor BA. Now, this factor B, as the norm remains, is going to join back. And that is how you have the formation of this complex C3b and B. Now, C3BB this is going to mediate the cleavage of factor D. Factor D is going to form factor DA and DB. So I'm sure this is getting complicated, but then this is it's the same information. If you see factor D getting cleaved into one larger factor and one smaller factor. Eventually, we have the C3 convertase formed again, but now the C3 convertase formed is C3b and BB. So cleavage of the factor B products and cleavage of C3 products. So make of C3 convertase in classical pathway is C4b2A, but make of C3 convertase in alternate pathway is C3b-BB. Okay, so again, but from here, the story is simplified because it's the same. C3 cleaving, this is going to join to this if it's the alternative pathway. We have the C3b molecule joining further, associating as C3b, BB, 3B, forming the C5 convertase. And once C5 convertase form, we have the formation of C5 small b joined by C6, 7, C8, C9 into a membrane attack complex. Now look here. This might make it easier for you. C3 spontaneously cleaving into C3b and C3a. So C3 hydrolysis spontaneously into C3b fragment, which attaches to a foreign substance. Now, what if this foreign substance is not it's not getting attached to it? If it is not getting attached to it, this C3b fragment disintegrates, right? But if it gets a chance to attach, this remains active for a longer time, which allows sufficient time for factor B to join the complex. Now, as factor B joins, in presence of factor D, the cleavage of factor B happens, and what remains with the C3b and foreign substance is C3b, BB. BA leaves the complex. Now, this forms the C3 convertase. C3 is now getting converted, and this complex is stabilized by another factor called factor P or properdin. If this properdin is not present, the C3BBB disintegrates so the half-life of this complex is around 5 minutes. So within 5 minutes, the concentration of this complex is halved. In the next 5 minutes, the concentration is further halved. So that's why we have half-life estimation of these products but so because if so half का half होने में 5 मिनट लगा, इसका half होने में फिर से 5 मिनट लगा, but then it's an infinite possibility, right? So which means essentially within 5 minutes, the half-life or the half of the concentration of it is already cleared off. But the association of factor B stabilizes the complex stable enough for it to have some time to leave C3 into C3A and C3B. Some of the C3B can associate back to it and form the, okay, so this is a typo on the book, into the C5 convertase. Okay, so now the C5 convertase, as the name suggests, C5 को cleave करता है C5B और A में और membrane attack complex बना देता है. So binding of properdin stabilizes the C convertase and convertase generates C3B. Some binds to C3 convertase, C5 convertase, C5 binds to antigenic surfaces. Okay. Now one more fact that why it's not like, so there is no specificity here. We are saying that this is binding to the foreign substance that makes it remain active for a longer time. What if it binds to the own cell, the mammalian cell? So in fact, it does bind to the mammalian cell, right? But this binding is not sustained for long enough. Why so? The host mammalian cell has high amount of sialic acid. In presence of this sialic acid, this C3B binding is inactivated, or the C3B molecule is inactivated. Now, since the pathogens and some lower phyla animals, they are not going to have this high amount of sialic acid, and that's how the binding of C3B is sustained to the foreign substance is sustained for long enough for factor B to come and associate with. And the binding of this factor B to this complex is magnesium dependent. So just some information for you to further munch on. Okay. Now, if you look here, there is no involvement of antibody molecule in the entire process of activation, and that's why this is an independent of antibody molecule pathway belonging to the innate immune system. So what remains? What remains is the lectin pathway. Now, as the name suggests, in lectin pathway, some sort of carbohydrates or some sort of lectin molecules must be involved. The mannose binding lectins. So these binds to the foreign surfaces. What happens? The foreign surfaces or the pathogenic molecules have high levels of expression of, say, the mannose or some glycoproteins or some carbohydrates on its surfaces. Now, these can be bound by these mannose binding lectins, which are present in the circulation. So, mannose binding lectins are acute phase proteins. Acute phase proteins, so these are released during the process of inflammation. So, in the presence of these acute phase proteins, MBL, they bind to the mannose or glycoproteins on the surface of the foreign substances. So this is very similar to the C1q binding to the antigen-antibody complexes. But again, there is no involvement of antigen-antibody complex. So this is also a part of, so this is also a part of, the innate immune system. So as this MBL binds to the foreign substance mannoses, there is association of MASP1 and 2, which are some MBL associated proteases. So just how we had serine proteases here, there is some other set of proteases that associate with MBL that is bound to the mannoses on the foreign substances. So again, since there is a protease that is involved, protease means it's going to cleave something. So it's going to cleave the C4 and C2. So if you look here, the cleavage of C4 and C2 and the eventual association of it into a C3 convertase. So the make of C3 convertase in your classical pathway as well as in your lectin pathway is the same. It's just that over here we had antigen antibody molecules being bound by C1 molecule, C1 proteins, but in here we have in the lectin pathway, we have the involvement of mannose binding lectins that are getting associated with some other proteases to eventually bring about the cleavage of the same set of complement proteins, and rest of the just this much is different. The binding, the initiation molecule is different. All the other things that follow in line from classical and lectin pathways is just the same. Okay. Now, it is important for us to understand that the complement activation is a tightly regulated processes, because if it's not the case, then there can be many target the host cells can also undergo suffer a lot of loss, or there can be indiscriminate cell lysis and death of maybe some important cells, right? So it's a very tightly regulated processes. A lot of information again. So there are multiple proteins that can regulate the complement system. So all the names that you can try and munch on. Let's focus here and try and understand how the regulation part is happening, right? So there are multiple points at which regulation can occur. First is before the assembly of convertase activity. So before the C3 convertase is formed, there can be certain molecules that is preventing the assembly of this convertase, such as C1 inhibitor molecules or C1IAB that binds to the C1R2S2 and causes to dissociate from the C1Q. So any black thing that you see here represents a regulatory molecule. So which regulatory molecule have they discussed? So they say that C1INH can bind to the C1QR2S2 and cause the C1R2S2 to dissociate from the C1Q part. So this was the protease, right? So if the protease gets dissociated, this cannot convert or cleave the C4 and C2 molecules, and that's how the system is checked for or it is regulated, right? But or in the next step, the next possible checkpoint or regulatory checkpoint could be where the cleavage of C2 and C4 had to happen. So there can be some factors which can either bind to this C4b fragment, such as C4b binding protein, C1 CR1, which is a receptor of complement proteins, or MCP. So these are all different inhibitor molecules that can actually bind to the C4b fragment and prevent it from getting assembled into a C3 convertase along with the C2A molecule. It is also possible that there can be another factor called factor 1 that binds to the that cleaves the C4 molecule. Instead of C4A and B, it cleaves it into C4C and C4D. So this is alternate cleavage that happens, and if we have C4C and C4D, the end result is the same, that the C3 convertase molecule is not formed. And so all this is happening for your classical pathway and the lectin pathway. The other possibility is in their alternative pathway. So in alternative pathway, similarly, C3b can be bound by some inhibitory proteins such as CR1, MCP, or factor H, or we can have involvement of factor 1 that cleaves the C3 into different component, different than what is required, the C3b, and eventually the C3 convertase assembly fails. So if the convertase has also, this is prior, all of this was happening prior to the preventing the assembly of C3 convertase. If the convertase has assembled, the next check that can happen is that the C3 convertase, so this convertase is dissociated by certain other factors, right? So which all factors? C4b-BP, binding proteins, some complement receptor, a factor called factor H, or DAP. So this dissociation, so convertase बंद तो गया था, but कुछ factors आके इसको bind कर गए, and that's how the convertase is dissociated, and the C3, C4b or C3b, they are cleaved differently by the factor 1, so that the future assembly of the convertase is also prevented. Or finally, if the convertases have formed and the regulation at the level of assembly of convertase also fails, there can be regulation at the level of formation of membrane attack complex. So the S protein, this prevents the C5b67. So क्या हो, C5 convert होता था, C5 convert होके C6 और C7 join किया, उसके बाद C8 को join करना था. But S protein, if it comes and bind, C8 fails to join. And if C8 fails to join, C9 will not join, and eventually polymerization of C9 is not going to happen. No membrane attack complex is formed. Other possibility is that C8 has also joined, but now comes the inhibitor, HRF or MIRL. This prevents the polymerization of C9, right? And if polymerization of C9 fails, again, the same consequence, the membrane attack complex cannot form, right? So again, different names of it, more details about how it prevents all of it, and we just saw how, you must understand how the regulation is happening and try your best to mug up as much as possible. Okay. So activation part of complement proteins has been discussed. We understand that they need to be regulated. The regulation part we have discussed. Now let's discuss the final part of it, that what all biological effector response can be brought about by binding of complement proteins. So one thing that we just saw is cell lysis, which involves C5b to C9 to form a membrane attack complex. The other effector response is that there can be a trigger of inflammatory response. There can be opsonization of particulate antigens by increased phagocytic uptake of them. There can be neutralization of viruses, and this part is it's an exclusive function of complement proteins, which is solubilization and clearance of immune complexes, and it's a very important part of your overall immune response, right? So as important as it is for the body to be able to respond to an immunological challenge, it is then very important for all the effector molecules to be cleared off from the system. Because if clearing fails, persistent upregulation of immune response is equally harmful to the body, as is the failure of an immune response, right? So let's just go one by one. So for any of the biological effect to be mediated, we understand by now that there has to be binding or involvement of some sort of receptors, right? Because complement proteins also in itself, they are bringing about cell lysis, but if you look here, so if it has to mediate an inflammatory response, it involves a lot of cells. It requires a lot of cells to participate in the process. And for any sort of cell to be participating in response to certain proteins, the cells have to have some sort of receptors, right? So we do have the biological effector response because of the complement proteins. It is facilitated by binding of the complement proteins to specific receptor presented on the cells of the immune system. And again, there is a list of it. So if you look here, receptors CR1, CR2, 3, 4, 5, 3, 4, 5, and different sort of receptors, right? So the name of it basically is denoting which complement protein fragment or which complement protein this can bind to. The major ligands for it, the activity that is triggered by binding of it, and on which particular cells the receptors are present. So one thing that we have talked about is the cell lysis part of it, following it up with the inflammatory response that is triggered because of the complement proteins. So this inflammation is mediated by all the smaller fragments that was generated as a product of cleavage of your complement proteins. So we have the larger parts participating in the cascade reaction, and the smaller part we say that they diffuse about to bring about inflammatory response. So these smaller parts are actually called as anaphylotoxins. So what is the criteria for something to be called an anaphylotoxin is that it should be able to bind to the receptors on your mast cells and basophils and induced degranulation of its content. So so because of the binding of the anaphylotoxin to the mast cells and basophils, there is degranulation of histamine and some other pharmacologically active components. Once these active components are coming about in the tissue, they mediate smooth muscle contraction. Smooth muscle contraction, which means the blood vessel smooth muscles, because of which the vascular permeability increases. Now, because of increased vascular permeability, we have leukocytes chemotaxis. In this process of chemotaxis, the anaphylotoxins can also participate, and different association products of the complement pathways can also participate. So C3A, C5A, C5B, 6, 7. So these are all mediating the chemotaxis, which means that the process of extravasation or marking the site where the leukocytes have to reach, they are all mediated by the complement proteins, and eventually at those sites, extravasation is permitted. So if you see here, this entire immune inflammatory response is actually mediated by the anaphylotoxins, which is nothing but the complement cleavage products, right? So the complement proteins in itself. So that's how the complement proteins can mediate an inflammatory response. The other response that they can bring about is opsonization. So what do I mean by opsonization? Opsonization refers to this process wherein there is some sort of coating of your pathogen molecules, and because of it, the phagocytic uptake of these particular pathogens become easier, and they are more likely to be eliminated of the system. So in opsonization, the major opsonin is the C3b molecule that is formed. So if you remember from the diagram, we say that some of the C3b uh, comes and binds back, but then what all is this C3b that goes out about does? So C3b, so the most potent anaphylotoxin is C5a. And C3b mediates most of the other functions, opsonization and neutralization, and complex antigen-antibody complex clearance. Most of these functions of the complement system is brought about by C3b. So C3b coats the immune complexes and the particulate antigens, which means, so look over here. So there's a bacteria. This bacterial antigen is bound by the antibody molecule. This antibody molecule can be bound by the C3b molecules, or independently also we can have C3b binding to the bacterial surface. Now, the phagocyte, they have receptor for C3b. So C3b pulls the bacteria along with the antibody molecule, which are also bound by the antibody molecule, towards the phagocyte. The C3b pulls the bacteria towards the phagocyte, and eventually the antibody also comes and binds to the Fc receptor on the phagocyte. So even if this thing was not there, there could be a phagocytic clearance of the bacteria, and that process is so it's not, so that is opsonization that is directly brought about by antibody molecule, but binding of complement protein further enhances this. So this is opsonization brought about by complement proteins. And if directly so let's just say that this is not there, instead of phagocyte, it could be an NK cell. So there's involvement of NK cells and the involvement of antibody. So that part was the ADCC that we have just discussed. Okay. So once there is binding of it, there is a quick formation of a phagophore, and eventually the bacterium gets phagocytosed. So antigen coated with C3b This and the interaction of the participating complement receptors on the phagocytes facilitates the process of opsonization. The other possibility is that C3b acts as an adjuvant and targets the antigen directly to phagocytosis. So what does that mean? That so maybe the bacterium is smaller in size and it's not um, pathogenic enough or antigenic enough. So if it's not antigenic enough, there is going to be no immune response. But because of binding of complement proteins on this small non-antigenic bacterium, this acts as an adjuvant. Adjuvant means it has enhanced the antigenicity of the bacteria. And now it is easier for the phagocytes to identify it and quickly phagocyte process so that there is clearance of the initially non-antigenic bacteria. Okay, the last two functions, which is the neutralization of viral infectivity, wherein the C3b, they aggregate the viral particles. So there are maybe the viral particle that are present extracellularly. C3b is going to bind to the viral particles and eventually it is going to bring about all the small particles together into a aggregate, right? Now, wherever there is an aggregation, the size increases, which means it is easier for the phagocytic clearance of it to occur. So this process is known as neutralization. Why neutralization? Because in the process of it binding the viral particles, it forms a thick coating on the viral particles that neutralizes the infectivity of the virus and prevents it to attach to the host tissue or cells. And lastly, the process of clearance of your immune complexes, wherein if and how was it identified that the process of immune clearance is very important? That there is a disease which of late has gained some sort of enhanced attention because of a celebrity diagnosed with it, systemic lupus erythematosus. So what happens in systemic lupus erythematosus is that your immune complexes are persistent in the circulation. So because of which the the mechanism or the response which was supporting the body in clearing the pathogen, because of its prolonged persistence, there is an hyper activated or chronically activated immune response which starts causing inflammation in the so areas which were not initially having any sort of infection, but because of prolonged activation of inflammatory response, there is now damage to the tissue. So that sort of that this is what happens in your systemic lupus erythematosus. This is a disease which is associated with persistence of immune complexes. And it was identified that in individuals with systemic lupus erythematosus, SLE, the complement receptors are deficient. So the complement receptors, right? So the receptors which were binding the complement proteins. If you look here, the receptors binding to the complement protein is going to clear, say, your antigen antibody complexes also, like here. So there we were talking about clearance of the bacteria, and over here, so this is insoluble immune complex. There is a complement activation, and C3b comes and bind the Fc region of the antibody. This this has to now bind to the complement receptor on erythrocytes. Again, as we were just discussing a couple of minutes ago, that the erythrocytes are having the complement receptors. This eventually goes to the liver and spleen, where the erythrocyte is stripped of this antigen antibody complexes, and now the Fc receptors on phagocytes are getting a chance to. So the antigen antibody complex is pulled into the phagocyte via the Fc receptors. So via the complement receptors. So if complement receptors are deficient, this entire process is compromised. So there might be there might be the coating of soluble antigen antibody complexes by C3b, But because the receptor is deficient, the C3b is not able to bind either to the erythrocytes or to the phagocytes, and that's how the immune complexes are persistent in the system. Okay, because, so we just discussed that the coating of soluble antigen antibody complexes by C3b facilitated to bind to the complement receptors in the erythrocytes. This is eventually carried to the liver and spleen, where the antigen antibody is stripped off from the erythrocytes, and then the phagocytic clearance of it happens. Okay, so with this, we complete our discussion with complement proteins. Now, if there is any doubts for this, we can take it up in our next class along with the notes for this. Uh, there is some straightforward topics that we uh, might want to discuss if required. I will be uploading the topics into the notes section onto the Google Drive, and we will not waste much time on discussing all of it, but just an overview. And if there's any doubt with from the note, that part can be taken up in the following class. So in immunological techniques, the principle that underlines all of these techniques is that there is a immune interaction, involvement of antibodies to the antigen. Now, what is the antigen? Why is it a technique now, right? So technique is to facilitate some sort of biological study or to identify some sort of biological analyte in your say pathological in pathological setups, right? Where you want to identify some disease causing pathogen, if it's present or not, or maybe which pathogen is responsible for whatever disease manifestation is happening. Yeah, we want to say purify some sort of protein, right? So all those things, that any sort of protein that either has to be identified or purified, or something has to be distinguished based on what is a protein marker present on something. So any sort of protein can very theoretically also and very imaginably, we can call any protein as an antigen. And if what binds to an antigen is a specific antibody. It is possible for us to specifically engineer an antibody molecule against a particular protein. Now, once we have antibody for a particular protein, this can be used for all the different purposes that we just talked. So, how much antibody is bound to a surface, per se, can give you an idea of how much how much quantity of a antigen or the protein of interest is present in a solution. Or if I have to say pull out a cell which has a particular protein, we can mark that cell with certain antibodies against the protein present on the cell, and then pull out the antibody along with the cell and antigen complex by some methods of precipitation, say, or some sort of column purification, say. So the bottom line is all of the immunological techniques, they require a interaction of antigen antibody, and that is why it is coming under the umbrella term of immunological techniques. So the techniques that are primarily used in day-to-day processes, which are very popular immunological techniques, it's ELISA, electrophoresis, immunohistochemistry, immunoprecipitation flow cytometry, and immunoaffinity chromatography. So ELISA is mostly used to detect an analyte. It involves your enzyme as well as your immunological interaction. So if a particular analyte is present, the antibody essentially gets a chance to bind to it, and the antibody is further conjugated to some sort of enzyme that cleaves or converts a chromogenic substance into a colored into its colored form, right? And this color can be quantitatively measured. In immunoelectrophoresis, so electrophoresis, you all have must have studied about it, the agarose gel electrophoresis, or SDS, where some sort of uh, isolation or separation of certain genes or maybe some proteins are involved. So, electrophoresis is essentially used for that. But now, if we bring in the binding or the participation of antigen antibody complex, so we can identify particularly that, say, which protein, if a protein is present in a particular band of if my protein of interest is present in this particular band that I see, right? Using special antibody designed against the protein of interest. Immunohistochemistry or immunocytochemistry, wherein we can identify certain markers present on these cells or present in a tissue. So tissue, if it's a tissue, it's immunohistochemistry, and if it's a cell, it's immunocytochemistry. The idea is we use some antibodies to identify if the protein of interest is present in the tissue or a cell. Immunoprecipitation is similar, that if there is a soluble antigen, I can incorporate some antibodies in the solution and allow the antigen antibody complex to settle down in the form of precipitate, which can eventually be isolated to isolate the antigen. Flow cytometry. So flow cytometry involves analysis of multiple cells. And how do I identify that which cell is the cell of my interest? Again, based on some sort of proteins. How to mark those cells is by using specific antibody against these proteins, which can serve as a marker for my cell of interest. And lastly, immunoaffinity chromatography. So immunoaffinity chromatography is wherein I am interested in purifying some sort of protein. So we have a chromatographic column, right? So chromatography can again be of multiple types. Just understand that there is some sort of a matrix which supports the interaction or binding of my protein of interest. Either it allows binding of the protein of interest because the column, this matrix, has the antibody which can trap my proteins of interest, or the matrix can be such that it allows my protein of interest to come into the elute or the flow through, from where I can again use some sort of antibodies and precipitate out my protein of interest. Right, so these are some basic immunological techniques. Consider it a much more straightforward topic to understand, and I'll be sharing the handout for the techniques. And if there is any doubts from this section or from the section that we just discussed, so there we can take it up. Okay. So with this, we can close our today's session, and our following sessions will be revolving more so about the innate immune system. And so one thing that we have not discussed in sufficient details is the MHC component, the MHC complexes. And finally, we can conclude our discussion with immunodeficiency and disorders of the immune system. I will see you all in the next class. Thank you.